We're on page 10 of the Unit 2 Biology Keystone Review. We've seen this question before. It was a short answer question on one of your exams. It says use the chart to answer the question. Molecule 1 and Molecule 2, biological molecule information. Molecule 1 is a molecule made mostly of carbon with two non-polar. As soon as you see non-polar, think of lipids. It has two non-polar chains and a polar head. This is probably a disaccharide. Okay, and it's also a component of the plasma membrane. Oh wait, maybe it's a phospholipid. Maybe it helps with waterproof coverings. These are little notes I made to myself, and these are little things that I expect you to do with your scrap paper that's provided for you on the Keystone exam. Molecule 2 is a polar molecule made of repeating units of sugar bonded to a phosphate and nitrogenous base. Sugar phosphate backbone, nitrogenous base. They must be talking about DNA or RNA. Wait up, it also stores genetic information. They're definitely talking about DNA and RNA, which are nucleic acids. So, which biological molecules are most likely represented by molecule number 1 and molecule number 2? Easy for you guys. It's molecule 1 is a lipid, molecule 2 is a nucleic acid. How did only 40% of students in Pennsylvania get this correct? It's the easiest question so far. I'm not even going to talk about it. Number 11, page 11. Which action must occur for an enzyme to catalyze a chemical reaction? I know that the word catalyze means to speed up. I know that enzymes speed up chemical reactions in living things. So, lots of words here, lots of vocab. Let's try to eliminate choices. A competitive inhibitor must first bind to an active site. That's not it, because we know that substrates bind to active sites. Oh, B, a substrate must bind to an active site of the enzyme. That's probably right, because the substrate is like a key, an active site is like a lock. Let's look at letter C and D. Substrate must bind to an allosteric site of the enzyme. It's actually the other side of the enzyme, like the dark side of the enzyme. We don't need to talk about that. Or a non-competitive inhibitor must first bind to an active site. Again, don't need to talk about that. I feel like this is kind of a difficult question because it's a lot of vocab related to enzymes, and there's already a lot of vocab related to enzymes, but somehow 65% of kids in Pennsylvania got this one right. You're going to get it right too, and that's that. Page 12. Lipase is an enzyme that breaks down lipids in the digestive system of humans. So we know it's an enzyme. We also have a clue because it ends in ASE. We're in the digestive system, and it breaks down lipids. It breaks down fats and oils. It functions best at a pH range of 4 and 5. When a person takes an antacid tablet to relieve heartburn, the antacid increases the pH to around 7 in certain areas of the digestive system. Which initial effect would most likely be caused by a change in pH? I drew a picture to help myself figure this out because we're talking about enzymes. So I drew this little enzyme table that we have. Lipids digested goes up over here, and pH goes over here. So at 4 and 5 is where this lipase enzyme works best at digesting lipids. If I take an antacid that pushes my pH up to 7, you're going to see a decrease in enzyme function. So that's going to mean that fewer lipids are digested. Let's see which multiple choice makes the most sense. More lipids digested? Nah. Fewer lipids digested? That's what my picture shows, but let's just double check my other choices. No, no. All right, this was again a level 2 question. If you really think it through, this should be easy for you, but take the time to draw the picture, guys. Don't just guess. This is easy once you draw it out. Unfortunately, most kids didn't draw a picture. There's only 36% of students in Pennsylvania got this one correct last year. And if we look at the explanation, B, lipid digestion decreases outside of the optimal pH range of 4 to 5. Take your time, read through your choices, use your scrap paper. The constructed response question for Unit 2, this is another way of saying the open-ended response question. Some things I want to take you through here. A group of students is given a sample of an unknown substance. The students are asked to gather evidence to determine whether the substance is living or non-living. The students take a small sample of the substance and observe it using a microscope. Good for them. Describe the 2 characteristics of the substance that could be observed to indicate whether it is living or non-living. Going back to your characteristics of living things, I gave you 12 of them. If you can remember 2, that'd be great. Remember 2 and start your answer with the question. And then describe an investigation that could be performed to classify the substance as living or non-living. Now, they give us the answer for this. If you can demonstrate a thorough understanding of the characteristics of life shared by all prokaryotic and eukaryotic organisms, describe characteristics that can be observed with a microscope, and describe an investigation to classify a substance as living or non-living. The response is clear, complete, and correct. That's going to get you the highest amount of points. If you can describe one characteristic of life or describe an investigation to classify the substances of living or non-living, that's going to get you 1 point. So what if we said the substance is made up of cells, or the substance contains genetic material, or the substance can reproduce, or the substance brings nutrients for energy, or the substance moves, or the substance grows and develops, or the substance, or for the investigation you could say, I could provide the substance with nutrients and check for growth. I could provide the substance with sunlight and check for growth. I could place the substance on a grid and see if it moves. I could count the number of cells and see if there's an increase in cells over time. I can touch the substance and check for a response. These are easy, guys. You just have to write something that lets them know that you know what the characteristics of life are. This is a 3-point answer. It's not a ton of writing. 3 points, so highest credit. If the substance is moving, it will indicate life. Movement requires energy, which can only occur in living things. So movement and energy are 2 characteristics of living things. If the substance grows over time, it is living. Growth requires cell reproduction, which only occurs in living things. They didn't even spell living right. And they got 3 points. How could they do an investigation that could be performed? The students could divide the substance into equal parts, put each part in a different container. When provided with time and proper conditions, the substance grew in time, it would be living. This growth would be due to the reproduction of cells through either meiosis or mitosis. This kid's an overachiever, but that's fine. This cell reproduction only occurs in living plants or animal cells. If it did not grow, it could be classified as non-living. This is due to the fact that all living things grow. Easy, guys. Easy stuff. Let's look at a 1 point. Look at it closely. See if it got cells. If it does, then it's living. 1 point. If it don't have cells or not showing that it's living, then it's non-living. Makes no sense, guys. 1 point. 1 point for grab a microscope and observe it very closally. 1 point answer. You guys can all get 1 point on that answer. I'm telling you. I have faith in you. Alright, so I'm not going to read through all the samples there. There's a lot of them. If you want to take a closer look at some of the sample answers, let me know. Alright, was that the last one? Okay, page 15. Use the graph and diagram below to answer the question. Explain how enzyme A acts as a catalyst in the reaction. Be sure to include energy and time in your answer. All right. So enzyme A, this is the active site, this is the substrate. They fit together like a lock and key. Without the enzyme, you see how long it takes for the reaction to occur. With the enzyme, you see that it's much faster. It's asking, how does enzyme A act as a catalyst? You're just going to describe how this enzyme speeds up the reaction by lowering the activation energy. All right, so explain how enzyme A acts as a catalyst in the reaction in respect to energy. Predict how changing the shape of the enzyme's active site would affect the enzyme's ability to catalyze the reaction. So by changing its shape would affect its ability to speed up the reaction. All right. That's that for session 2.